Great. So welcome back from a very short break. So as you all are aware, we're really keen on getting everyone ready to pitch to investors. So thank you everyone, first of all, for submitting your 90-second pitch with kind of short turnaround and very little instructions. For the most part, everyone did awesome as far as a first try, and you actually had the chance to decide who made it to the final based on your votes and some of our other internal kind of voting as well. So it's my pleasure to introduce this investor pitch session where the finalists will give a pitch in front of our panel. Is there a panel here? I can't see their faces as panelists.
Yes, Minnie is here.
Okay, so we recruited kind of a pitch panel that kind of represents different parts of like the investor spectrum, right? So we have Dennis from the Resi conference, so he sees a lot of investigators or investors. Minnie, who does more kind of like economic development type stuff. Prasad Sukara, who is part of the Angel Capital Association. And then Kwame Ulmer, who wears many hats. He's formerly from the FDA. He has his own consulting firm and is an investor, and also works as an investor in residence with Steph Davis in the Office of Translational Coordination. So I thought this was a good mix of different perspectives from different parts of the investor world to give feedback to our finalists. So I will hand it off to Marissa. Are you going to announce who's the order?
Sure. All right, everyone. So we have 5 pitching teams today. So Dr. Suneet Agarwal, Dr. Kevin Snook on behalf of the Bagwell team. My regards to Dr. Bagwell today and could not deliver his pitch. So Kevin Snook took over within mere hours of the pitch event. So thank you so much for that. Dr. Rebekah Gundry, Mr. Ray Liu on behalf of the Kim team, and Mr. Rob Bent on behalf of the Vanderslice team. So I think we will just go in that order. So Dr. Agarwal, are you ready?
Yes, thanks. Can you hear me?
Yes, we sure can.
So I cannot share a screen. Okay, maybe now I can. Great. All right, and just hopefully you're seeing that, and I should put it in a presentation type mode here somewhere. Oh, there it is down there.
That's perfect. We can see it and hear you well.
Okay, great. I'll start then. All right, so thank you very much for this exciting opportunity. My name is Suneet Agarwal, and I'm a pediatric hematologist and bone marrow transplant specialist. And today I'm really excited to tell you about our mission, which is to develop the first molecularly targeted drugs for lung and blood diseases called telomere diseases. So the prototype and the problem of the telomere diseases is a rare genetic condition that I specialize in called dyskeratosis congenita, or DC. And I'll exemplify the problem with a vignette of these 2 girls so dear to me, who I diagnosed with DC when they presented with failure of their blood system at the age of 18 months. So they underwent bone marrow transplant and they did well, but here they are at age 7 doing well, but underneath it all, their other organs were failing and they developed hypoxia and now needed lung and liver transplants. And unfortunately, neither of them survived. So this is my unmet need slide, and addressing this is my entire motivation. But why should it matter to you? So the telomeres regulate cellular aging in all of us, and dyskeratosis congenita turns out to be the tip of an iceberg, a part of a continuum of telomere diseases. They're all caused by germline mutations in genes regulating telomere maintenance, presenting at different ages of life. Telomeres are the ends of the chromosomes. They undergo attrition as we age. They're important for tissues to renew themselves. So people who inherit mutations in the machinery important for telomere maintenance have these so-called telomere diseases that occur over the lifespan, and there are no cures. The standards are tissue transplantation and supportive care. And we need a solution that provides systemic therapy that restores telomeres throughout the body. So what did we discover? By studying mutations in our patients, we discovered a pathway with unexpected specificity for regulating a key component of telomerase called the non-coding RNA, TERK. This was completely out of left field. We found, and others validated, that an enzyme mutated in DC and PF was responsible for productive maturation of the non-coding RNA, which controls telomerase levels. We then discovered and validated a target, PAPD5, that opposes this and proposed that inhibiting it would restore the balance and levels of telomerase, performed a high throughput screen and discovered novel small molecules that inhibit PAPD5. And so what do they do? So in multiple patient-derived induced pluripotent stem cells treated with vehicle versus drug, we showed that telomeres lengthened by hundreds of thousands of nucleotides over the course of weeks. And this is very unanticipated and unprecedented, manipulating telomerase with small molecules for the first time. So our competitive edge is that these are the first small molecules that restore telomere length in human stem cells. So this is just showing our hit, and it's published. But we've taken this in vivo, have optimized potency and other parameters, and these are supported by several IP filings that are owned by Boston Children's Hospital with clear priority in the field. Our regulatory path would be standard for a small molecule, and we would pursue bone marrow failure and dyskeratosis congenita as a lead indication based on feasibility for proof of concept. This path will take advantage of FDA incentive programs, including fast track, orphan disease, and pediatric voucher programs. The rapid follow-on indication would be pulmonary fibrosis, and beyond exit other applications, perhaps in degenerative diseases and aging. So given the time, I'm just going to focus on the market opportunity in pulmonary fibrosis, which may be more familiar to you, but even to this expert group, it may not be known that impaired telomere maintenance is causally associated with pulmonary fibrosis, with hundreds of articles over 15 years. And this area is a hot area for therapeutic development by various pathways, but down here in the corner is telomerase, and there are 0 drugs in this box. So our intended use is pulmonary fibrosis patients with genetically confirmed telomere disease. This would be an oral drug, chronically administered. And there are thousands of US patients and more deaths than breast cancer from pulmonary fibrosis, and thousands of these patients have underlying telomere disease. As you know, there's existing drugs. There are 2 approved under breakthrough status costing more than $100,000 per patient per year, with $2.4 billion in sales, another with $8.3 billion acquisition. And neither of these change quality of life. They just decrease the rate of decline of FVC, which is not symptom altering for any individual. So our approach has a non-overlapping mechanism of action. It's first in class and goes to the genetic underpinnings of the disease. Our team highlight Dr. Neha Nagpal, who drove the small molecule work, a fantastic biochemist and medicinal chemist, and therapeutic development team. And we received over $3 million in non-dilutive funding through numerous accelerator supports. Our team extends to a decade of work with patient advocacy groups. I serve as the chair of the medical advisory board and leadership in international consortia, conducting a multi-center clinical trial. So to summarize, we are tackling life-threatening diseases of children and adults without targeted or curative medical therapies. Our solution goes to the heart of the matter, and I'm still blown away that we're drugging telomerase for therapeutics. We seek partnership to advance a robust lead optimization program and enable first-in-human trials. Our strengths are listed here, and I'll just highlight that this is based on solid human genetics, both Mendelian and at the population level, that indicate telomerase is associated with longevity in the general population. So this provides a major upside in the burgeoning anti-aging space. So in terms of impact, please help us by developing the first targeted telomere therapies for fatal diseases of children and adults. So thanks again for the opportunity, and I'd love to speak to you further about working together.
Thank you so much, Dr. Agarwal. That was right at 5 minutes. I always love when that works out perfectly. Our pitch judges, just as a reminder to everyone, will provide feedback after all 5 teams have pitched. So if the judges are ready to hear the next pitch, please let me know, and I will turn it over to our next team.
When do we ask the questions? At the end, or?
Yes, Prasad, at the end. So we will hear all 5 pitches first, and then we can have a bit of a discussion with each team.
Okay, thank you.
Sure. All right, pitch judges, give me a thumbs up if you're done on your score sheets and ready to hear the next pitch. All right. All right, good to go, it looks like. So I will now introduce Dr. Kevin Snook on behalf of the Bagwell team. Are you ready?
I think I'm ready. Can you hear me?
I sure can.
Okay. Let's see if this works.
Yep, that works.
Sharing?
Yes. Are you able to turn your video on today, or are you not at a video?
I am at video, but it says I'm not allowed to turn it back on because it was turned off by the host.
Okay, Patrick, can you take care of that for us?
See if that works. There we go.
There you go, Kevin. You are in front of us whenever you're ready.
All right. Just minimize that. All right, I think I'm ready now. Yes, so I'm Kevin Snook from Actuated Medical. I'm lead engineer here in the program, and I'm speaking on behalf of Roger, unfortunately has COVID. So my talk today is focused on venous thromboembolism, or blood clots in veins, and specifically the legs, known as deep vein thrombosis, or DVTs. Now, this is a condition that can happen for many different reasons, but if dislodged, these can become stuck in the lungs, becoming pulmonary embolisms. It's a serious condition with a 25% chance of death. Now, there's potentially millions of people that can have this throughout the world. But this really isn't just about millions at risk and mortality statistics. DVTs are really very real, and they really change lives. My brother's sarcoma actually was found because of DVTs that were forming. And though he's cancer-free now, he actually continues to have DVTs and complications from this to this day. And he has to have compression leggings. He has difficulty walking. He takes blood thinners, and he's actually constantly concerned that he's going to leave his kids without a dad if he has an embolism. And you know, the real issue here is that current detection methods, they're only used after clots get big enough and dangerous enough to cause pain and other symptoms. There's not really any good tools to monitor these early forming clots and give an early diagnosis. So what we're doing is we're actually developing a wearable thermographic sensor technology that monitors subtle changes in the heat patterns in the leg due to blood flow. As you can see in this infrared image of a DVT patient, there's actually significant changes that you can see happen between a good leg and a bad leg. Now, this is actually within tenths of a degree, and this is a very symptomatic patient. But the thing is, you know, you can't really do a continuous IR image all the time for somebody. It's not an option. And so this graphene design enables a flexible sensor with very high resolution for small heat transfer patterns that happen with underlying veins. So basically, the single patient use device wirelessly alerts clinicians when flow patterns deviate above threshold. Would enable continuous bedside monitoring with minimal personnel and cost, which competitive tools lack. I'm going to show a data graphic illustrates here the flow status of an underlying vein over time in a preclinical work that we're doing. And in this occlusion study, the vein went from open to ligated, and you see there's a temperature pattern that shifted out of the normal within the circle. Now we're focused 2 stages with first several inpatient populations, looking at those, addressing that, and then going to larger expected market of home use. This is a much less controlled environment, but combined, the serviceable attainable market is estimated at just under $1 billion. And our current IP is based on a combination of licensed tech from our Penn State collaborators, filing patents to add to our current portfolio of 25 US and multiple international patents for other technologies in our company and trade secrets. We expect to achieve FDA clearance through a de novo pathway with clinical data. We expect that the sensor can actually result in savings of up to $28,000 per occurrence in the hospital based on reduction of thrombectomies. Now, where we're looking at right now is basically our plan is to exit to a strategic partner in 2026. Looking to get about $2.5 million fundraising to complete testing, validation, and clinical studies and achieve the FDA clearance by the beginning of 2025. Now, though ambitious, we're positioned for success. We're ISO 13485 certified and FDA registered, and we've actually transitioned multiple techs from grants to actual commercial market. And we currently several products on the market. And our seasoned team members have a track record of development success in medical, industrial, and defense sectors. We're backed up by a strong board of directors with experience and a keen focus in medtech. So in short, we have a wearable DVT sensor technology we believe will have a significant impact and give countless patients like my brother and their families peace of mind. So we're currently focused on the next steps and are looking for the investment to complete our V&V and clinical evaluations. Thank you for your time.
Great. Thank you so much, Kevin. That was just about 5 minutes as well. And your slides are really, really well done. I enjoyed that presentation. Pitch judges, once again, when you are finished with your score sheet, please give me a thumbs up and I will introduce our next speaker. And Dr. Gundry, just to give you a heads up, you are next in our alphabetical order. Great. All right, great. Looks like we are all set, pitch judges. It is now my pleasure to introduce Dr. Rebekah Gundry.
Okay, so you can hear me and see my screen and see me?
Confirmed.
All right. So hello, my name is Rebekah Gundry, and our GlidePack technology is going to change the future of biomarker discovery and precision medicine. Imagine a future where doctors can test a few drops of blood and tell you what treatment is best for you. And I imagine this because last year it took many tests for doctors to tell me that I have multiple sclerosis, but they could not tell me which drugs are best to manage my disease. So I started collecting my own blood on these blood spot cards so that someday I can fix this problem. The problem for MS, like 93% of all human diseases, is the lack of biomarkers for diagnosis, prognosis, and treatment. Without good biomarkers, disease diagnosis can be delayed, and treatment commonly proceeds by trial and error. A solution to this problem is to exploit glycans, which are coding structures made of sugars that encode information that are essential to life. Glycans code every major cell type in your body, antibodies and viruses, and proper glycan structures are necessary to maintain health, and incorrect glycans are present in every major disease. Glycans are responsible for live or die decisions. Your blood type is defined by the type of glycan that you have on your red blood cells. We know that if a patient receives the wrong blood type, they can die. That's why more than $3 billion annually is spent on blood typing. And this is just a single example of the importance of glycan analysis. Another example is prostate cancer. Shown here are data for 3 different analyses of prostate-specific antigen, which is a protein biomarker of prostate cancer. By measuring PSA with a specific glycan structure, cancer detection increases from 56 to 96% compared to free PSA. And this is all because the glycan structure present in PSA from cancer is a different arrangement than the glycan on PSA from normal patients. Despite their value, glycans are not exploited routinely in precision medicine. This is because they're challenging to study. There are no high throughput methods to prepare samples, so analyses are limited to a few labs. To solve this problem, we developed GlidePack, the first easy, standardized, high-throughput kit to prepare glycans for biomarker analysis by mass spectrometry. Starting with just a drop of blood, GlidePack will separate glycans from the proteins, then prepare and clean the glycans. The process can be fully automated with standard laboratory robotics. No specialized expertise is required. One user can process up to 384 samples in one day, and the samples are suitable for structural analyses. Using GlidePack, we can categorize rheumatoid arthritis patients at the time of diagnosis into 2 groups, those who will respond well versus not well to the first-line drug. This has potential to save money and health by helping clinicians select the right drug faster. Unlike other kits, GlidePak can discover new glycan structures and is applicable to a broad range of sample types. Overall, no current product matches the performance and capabilities of GlidePak. So glycans are everywhere. So GlidePak can be applied to any biological specimen type for any disease, ranging from cardiovascular to autoimmune diseases. The market size is millions to billions. Specifically, imagine if we could improve precision and accuracy of existing tests. With the PSA test, glycans add a 1.7-fold improvement to precision and accuracy. So applying that increase to the biomarker market takes that spending to just under 90 billion. Also, as biologic patents are expiring, biosimilars are on the rise. And while FDA approval already requires glycan analysis of biopharmaceuticals, there's an increasing demand for more detailed structural analyses, as standard approaches can miss novel, unexpected glycosylation events that impact function. The GlidePak sample prep kit is in production stage, and we and our partners are ready for a next stage investment of $3 million to integrate our kit with proven solutions from our commercial and academic partners that provide the robustness and speed for data acquisition and analysis that are necessary to transform our ideas into a true plug-and-play platform that can be installed in any laboratory. Our team has tremendous experience in biomarker development, glycomics, mass spectrometry, software development, and commercialization of similar products. Our prep kit will be commercially available next year. Then we will integrate this kit with new data acquisition and analysis solutions starting in 2024, and we will launch the complete platform in 2028. Granted patents protect fundamental aspects of the workflow, and others are protected by trade secret. Our commercial partners can manufacture in accordance with ISO standards so we can deploy as laboratory developed tests. Once adopted, our technology will improve diagnosis and clinical decisions regarding the best treatment strategies for patients and evaluation of the quality of therapeutic biologics. So in summary, glycans are currently an untapped resource for precision medicine. Help us change this by pushing forward a proven technology that makes glycan analysis easier and better. Thank you.
Fabulous. Thank you so much. I will give our pitch judges just a few more seconds to complete their scoring sheets, and we'll then introduce our next speaker. Pitch judges, how are we feeling about the time? Good. Excellent. All right. With that, I would love to introduce Mr. Ray Liu on behalf of Dr. Joshua Kim's team.
All right. Thank you, everyone. Let me go ahead and jump into slideshow mode. Okay. Can everyone see my slides?
We can, and we can hear you well.
All right. Thank you. Hi, my name is Ray Liu. I'm the CEO and co-founder of Vena Vitals, where we make a small wearable device that monitors blood pressure continuously. So blood pressure is one of the most important vital signs, but our tools today for monitoring it are really limited. We have the traditional cuff, but that only gives us snapshots in time, even though our blood pressures are constantly changing. And the only way to track these continuous changes currently is through an invasive procedure called the arterial line, but that requires an invasive procedure, which is painful and also introduces a number of potential complications. So at Vena Vitals, we've developed a technology that allows us to non-invasively capture all of these beat-to-beat changes through a wearable, flexible sensor that attaches up to the skin anywhere where we can detect a pulse. Now, our sensor compresses with each beat coming through, and we're able to translate this to a blood pressure measurement. The technology is protected by a suite of patents, 3 of which are granted and 2 that are currently pending. So our clinical studies currently ongoing are in the operating room comparing head-to-head against the arterial line, and we're able to show a near exact match to that invasive procedure. So the implications are profound because what we can do is number one, replace a invasive procedure that's currently still being performed more than 8 million times in the US alone each year. And on top of that, we can, because of our wearable form factor, we can actually bring continuous beat-to-beat monitoring beyond the hospital to new applications such as sleep, which I'll get into in a minute. Our current validation path has brought us to testing on over 100 subjects in the operating room head-to-head against the arterial line, covering a wide range of patient populations, including ages, BMIs, as well as different blood pressure ranges. And the results are extremely compelling. So the market for blood pressure monitoring is enormous, as you can imagine. And that's because blood pressure is used everywhere from the hospital to the home and to the now expanding wearables and digital health market. But we're not trying to boil the ocean all at once. And we're focused initially on two specific markets, first in the hospital and anesthesia surgery, and then in the clinical home for sleep monitoring. Now, on the surface, these might seem like two very different markets, but essentially it's the same product, because if you think about it, a patient lying on an operating table is actually very similar to a patient lying in bed. I want to drill deeper into the sleep apnea market because this is actually the focus of our NIH Catalyze grant, as well as our current fundraise right now. So apnea is one of the most underdiagnosed conditions, and it affects, for a male population, about a quarter of the population and 13% of females. That brings it to about 1 billion people worldwide who suffer from obstructive sleep apnea, with a prevalence where they have at least 5 events per hour when they're sleeping at night. And the market today, just for medical devices in this space, is already 8 billion with a 7% CAGR. But the implications are huge because patients who do suffer from obstructive sleep apnea have a 2 to 3 times higher risk of heart attack and stroke than compared to other conditions. And their life expectancy can be reduced by up to 10 years. So how does blood pressure relate to sleep apnea? Well, it really is the consequence of these apnea events. What you can see here in our chart is that after each apneic event, which is highlighted in blue, we have a corresponding surge in blood pressure as a result of the sympathetic activity and the body recovering from that loss in oxygen. When we zoom out from this view, what we can see is very profound. On the left is a subject who is a healthy subject without apneic events. On the right, this is a blood pressure variability profile for a subject who has been diagnosed with obstructive sleep apnea. And you can see how much stress this subject's cardiovascular system is being imposed upon due to all of these blood pressure surges. Now, as a company, we've only been around for 2.5 years, but we've really accomplished quite a lot in that short amount of time. We've raised over 5 million in funding, and we still have about half of that in the bank. And that covers us for our first market, which is the hospital operating room market. Right now, we're seeking an additional 1 million to be able to approach the sleep market, to be able to bring that market further up and accelerate that development and supplement our NIH Catalyze grant. As a team, we have a wealth of experience, including a number of exits, including to Vizient and Cigna. And also, we have experience launching medical devices to the market, including patient monitoring devices specifically. But this is really, we've never been as excited as we are today to be able to fundamentally transform something so important as blood pressure monitoring. And we hope you'll join us for this journey. Thank you.
Fantastic. Thank you so much. This is my favorite part of the annual meeting. It was last year and it continues to be this year, hearing all of this wonderful research and science. I have a background in biology and chemistry myself, so it's so exciting to hear the innovation, and I hope that everyone else feels the same way. All right, pitch judges, I will wait for your cue to move on to our 5th and final team's presentation. Great, thank you, Minnie. Everyone else, pitch judges, how are you feeling about time? Good? Great. All right, so with that, I will introduce Mr. Rob Bent on behalf of Team Vanderslice to give our final pitch presentation.
All right, hi everybody. Let me share this real quick. Okay. Oop. Hear me and see me okay? Got the presentation up? Okay. Hey everybody, we're really glad to be with you in the finals and have a chance to tell you a bit about Seven Hills. So Seven Hills is developing a platform of first-in-class small molecules that improve the effectiveness of a broad range of immunotherapies by activating cell adhesion molecules called integrins, which are essential for generating an effective antigen-specific immune response. As we age and acquire certain comorbidities, such as high central obesity, diabetes, and smoking, our integrin function decreases, resulting in higher risks for infection and cancer, as well as poor performance of vaccines and immuno-oncology drugs. Our mission at Seven Hills is to leverage the novel technology of integrin activation to deliver the promise of immunotherapy to all patients worldwide, regardless of age or immune status. Our solution is a revolutionary treatment regimen combining any commercial or developmental immunotherapy with our orally delivered compounds, normalizing integrin function to increase cell adhesion and improve immune responses. Here's a bit of data showing the difference that our lead phase 2 ready molecule 7-HP349 can make to both cellular and humoral immune responses. On the left, you see antibodies against H1N1 influenza produced by mice vaccinated with Fluzone. Adding 7-HP349 generated a large dose-dependent increase in antibodies and the rate of protection, or seroconversion. On the right, you see survival curves for mice bearing extremely cold melanoma tumors. This tumor model is so non-immunogenic that PD-1 therapy has almost no effect. With CTLA-4 blockade, you get about 25 to 30% survival. However, with 7-HP349 added on, we can almost triple survival. Our integrin activators have many potential applications, but we see 3 initial markets. The first, which is related to our Catalyze grant, is improving the utility of cord blood in stem cell transplantation, offering sales of around $200 to $400 million per year. Our 2 other initial markets, geriatric influenza and PD-1 resistant solid tumors, offer annual sales of over $1 billion each. Our key markets are competitive, but we're well positioned with strong differentiation. In flu, the majority of innovation is focused on creating a universal vaccine to avoid the yearly reformulation required to match the vaccine to the viral strains in circulation. However, a universal vaccine would not address the poor responses seen in the elderly population, making 7-HP349 perhaps the perfect partner to augment whichever universal vaccine eventually wins out. In cancer, the PD-1 resistant space is primarily focused on intratumoral agents, aiming to boost responses to checkpoint blockade. While the biology is solid, accurate intratumoral delivery has proven to be a significant hurdle, resulting in poor clinical outcomes and increased toxicities. Delivering these drugs also requires specialized facilities and personnel, increasing treatment costs and lowering patient access. 7-HP349 may offer the same benefits to more patients for a lower price with no added toxicities. Our IP estate is broad and pioneering, effectively cornering the market on integrin activation for immune stimulation out into the early 2040s. With less than $7 million in angel funds and about the same in NIH grants, we created a novel biological concept, identified a lead molecule, filed an IND, and completed a first-in-human phase 1 trial. We're now poised to hit multiple value-creating milestones highlighted in discussions with large pharma as thresholds for deal-making. We're raising $10 million to first execute a quick geriatric flu trial to demonstrate biological proof of concept. With another $25 million, we'll execute a second trial in PD-1 resistant solid tumors that will show clinical proof of concept. A positive readout in the flu trial may allow out-licensing within 18 months, and success in the cancer trial may enable a larger out-licensing or M&A deal within 3 years. This is our illustrious management team, a proven group of seasoned executives and developers with true molecule-to-market experience. Together, this core team has over 50 drug and medicinal product approvals to their credit. Our scientific advisory board comprises key opinion leaders in integrin biology, infectious disease, and cancer immunotherapy, providing insights to optimize our development pathway and product positioning to maximize our value in our target commercial markets. In closing, we welcome you to join us on our journey to deliver the promise of immunotherapy. We're seeking $10 to $35 million to reach near-term value-creating milestones with our first-in-concept technology. With success in the clinic, we may be able to offer investors opportunities for liquidity within 3 years via out-licensing for multiple billion-dollar indications. Our compounds may offer lower treatment costs, better access for patients, and improved responses to a broad array of current and next-generation immunotherapies. Thank you.
Great, thank you so much. 5 wonderful presentations, and I'm really excited to invite our pitch judges now to provide some comments and use the last about 20 minutes of the session as more of a workshop to provide comments to the speakers, but also provide a bit of context for each presentation and those listening to make this a valuable session for everyone. So pitch judges, whoever would like to go first. Let's start with Dr. Agarwal's presentation.
Yeah, Dr. Agarwal, interesting presentation. And so did you identify this particular enzyme pathway? Anybody else has worked on it before? Or, and also you have this inhibitor you're talking about. Is it a small molecule? I suppose it is a small molecule. Do you already have the bioavailability? Is it oral or IV? I'm sorry, I'm asking all this so that you can give a succinct reply. Thank you.
Sure, happy to. Thank you for the questions. So we identified this pathway in regulating human telomere biology, validated genetically. It was rapidly validated by other leaders in the field also. So the biology around it is solid in multiple systems by loss of function. And the molecule that we discovered, we have improved it, and it is orally bioavailable and shows efficacy in a mouse xenotransplantation model, which is exemplified in our published work, but not the molecule itself.
What is the model? Is that for a DC or something else?
So human telomere biology is distinct from the pathways that are affected in rodents. So the xenotransplantation model is creating telomere deficiency in human hematopoietic stem cells, introducing them into immunodeficient mice, and then treating the mice orally and reading out molecular phenotypes on the human hematopoietic stem cell.
I see. Okay, thank you.
Thank you.
Hi, this is Minnie Marwaha, one of the judges. Dr. Agarwal, thank you so much for your presentation. I think perhaps I was invited as a judge because I'm unlike most of you at the table. I don't have a PhD, I'm not clinical, but I've worked in healthcare for a long time as a hospital administrator, so I wear a different hat. Dr. Agarwal, just one piece of feedback that I had, and I see this a lot with pitches, and so unfortunately I'll pick on your presentation today, but this does apply. I encourage all 5 companies to think about this in reflection for their own pitches. I got lost with who your target market is. You started with pediatrics. You showed us a wonderful need and a compelling story at the beginning with the 2 twins. But by the end of the presentation, you began talking about pulmonary fibrosis, which is traditionally an adult, potentially geriatric associated condition. So as an investor or someone reviewing the pitch, the details got jumbled for me in terms of, okay, who are you actually serving? Who are you actually solving for? So that would be kind of one piece of feedback that I have, is just stay consistent from who you start with and how you're ending your presentation.
I appreciate that very much. Yeah, I actually have received that feedback before, and I think that part of it's the balance between trying to compel in a rare disease indication versus trying to show potential broader impact. But I appreciate that very much, especially for a small presentation or short presentation, keeping the indication consistent. Thank you.
Yes, I would have a kind of a general comment as well, in that you got to get your story down, and then you've got to get your market down. So what I'm hearing is that there's a lot of great new technologies, and the telomere diseases, et cetera, but you really got to pick one, because the reason is that each market has its players. And so there might be, VCs and PE and angel groups and family offices in a particular rare disease or whatever. But as you go from disease to disease, you're going to find different players and different investors. So when you're presenting pick a card, any card, I can do several diseases. You're basically, doubling and tripling your work when you're saying, well, I can go after this market, I can go after this market, I can go after this market. They all have different players. They all have to be mapped. So I always say, if it's an early stage company like yours and it's a potential platform like some of these other players, pick one. And then go as far as you can with that one. And if you're not getting the traction you need, then you can pivot. But trying to go after all three is really going to elongate the effort and defocus what you're trying to do, which is get a licensing partner or find some early stage dollars.
That's great advice. Thank you very much. Appreciate it.
Great. Kwame, any last comments for Dr. Agarwal?
Briefly, it didn't jump out at me, your next step and your raise. I may have missed it, but that's something that I missed, at least in your presentation. And I agree with the other comments about the rare disease, picking that because that's where you have the most data, the most traction. And oh, by the way, depending on how it's priced in the market, it may be a lucrative business, because you can be a rare disease and still be a very big business in terms of revenue, depending on how it's priced in the market. So thank you.
Thank you very much. Sounds good.
Fantastic. All right, pitch judges, let's move on to Dr. Snook on behalf of the Bagwell team. Any comments?
Yeah, I actually, once again, because in the world that I live in, which is connecting early stage technology assets with different partners around the world, I always find it's, we have to really stay away from just talking about the science and just talking about the, how deep you can go with solving a particular problem, that it's really good to be able to net out exactly what you do in the first, 5 seconds. So when I was looking at, a wearable thermal sensor for detecting DVT, I really like that, because I know exactly who this person is. I know exactly what they're doing, what their company's doing. And we know there's a problem in DVTs in elderly, and it's a biomarker for everything from lymphoma to potential pulmonary embolism. So I really urge all of the CEOs in early stage companies, how do you net out what your value is in a simple 5 to 8 words, a wearable thermal sensor for detecting DVT? I mean, how simple can you get? But it describes exactly what you do. After you get that simple sentence that can describe who you are and what you do, then you can go into your elevator pitch, get a little deeper, and then you can start talking about how your story developed with your IP, etc.
I agree with what Dennis just said, this Prasad Sunkara. But I would also add, what are the savings in terms of pharmacoeconomics, or with competing products or existing products would be very valuable too.
Okay.
Dr. Snook, I really enjoyed your opening story, the personal story. I think all 5 teams attempted this in different ways. And, please continue leaning into that. A personal story can really pay off tremendously, especially when you're this early stage and potentially talking to investors that don't know the science. So just connecting it back to the human and the person and how this actually impacts people's lives can really be profound and can change a person's choice to continue listening in the meeting and potentially asking you for a follow-up meeting. So good job on that. I thought you guys did the best at that. And then I would just offer one piece of feedback, and this I would say for all the companies that participated today is, I think you have to know who your audience is, and, if there's scientific, definitely include the graphs, include the acronyms, the nomenclature. But when someone's not scientific, you have to kind of almost have 2 versions of the presentation. One that's really like layman's terms, something that your teenage son or daughter could understand, and then another version that's the scientific version, and then everything else can go in the appendix. So when I was listening to your presentation, Dr. Snook, I was thinking, gosh, I'd really love some more visuals instead of the graphs, because it was too much for me to digest in such a short period of time as a layperson. And sometimes that's best done by like even just mapping out a patient journey to say, here is what the current problem state is, and our solution will create this ideal future state. And just some visuals, animation, anything that just can paint a picture versus showing it through data.
Okay, thanks. Yeah, that's good to know.
I'm going to ask a question just to confirm my understanding. And we're talking about, Mr. Bagwell, your presentation. Am I understanding this right? Or we're on a different presentation?
Yeah, so this is Kevin Snook. I'm speaking for Dr. Bagwell. He actually unfortunately is sick, so I'm speaking for
Got
him.
it. And is it my understanding, and please, there were a lot of presentations and I couldn't take all the notes. So is this the one where Dr. Mulhill is the CEO?
Yes, that's right.
Okay. So you send a signal when someone other than the CEO is presenting. You had a great, so the person presenting who was going to present isn't the CEO. He got COVID, totally understandable. So I would just immediately hit the narrative of this is why the CEO is not presenting, and this is why, because most investors want to be meeting with the CEO of the firm. That's my only substantive comment. It may seem stylistic, but there's substance behind it, because for early stage companies, generally the CEO is pitching. And if it's not, it's a signal of the level of commitment to raising capital. And I'll justify it as well. Most investors think in terms of the ability to raise, run, and recruit. And if the CEO isn't there, it's a signal of their commitment to the ability to raise. And that's my comment.
Yeah,
Okay.
I actually thought I heard you say that the reason you were there is because your CEO got COVID. So,
well, actually both. They're so Dr. Bagwell and Dr. Mulvihill are married, and they actually both have COVID.
Oh,
it's not
okay.
funny, but we get it now. Yeah.
All right, thank you so much for that round of comments. Let's move right along to Dr. Gundry's presentation. Pitch judges, who would like to start our comments?
As long as I'm talking, I'll be happy to start off. Once again, we get to this tagline thing, and we have to make it simple and we have to make it understandable. And with Rebekah's, I think it was the future of precision medicine. But when I really started when we really started listening to her presentation, it really turned out that they're really, evaluating glycan biomarkers for, diagnosis for disease, which, I find really that's kind of exciting, because the more biomarkers we have that we can prove efficacy and really dial in on diagnosing specific features of diseases, it's just, such a great place to be. And so, when you go into the future of precision medicine, what does that really mean and who's your audience? But when you're saying, hey, we're really figuring out, glycobiomarkers for disease diagnosis, now it's a diagnostic. They're doing they've got a new category of biomarkers, and they're having really compelling results across the board on many diseases. So I applaud what you folks are doing. And those are just some comments. Once again, try and keep it simple, but also direct on what the heck your technology does.
I was a little confused. I was quite enthusiastic when you said you can actually do a better job with measuring PSA, including glycan analysis. But when you then started talking about GlyPack, it looks like are you trying to do a CRO, analyzing antibodies for the glycan structure? So I think you need to focus on one area, or at least you can say, look, we want to also do a fee for service, or we want to sell these kits, but at the same time, we want to also develop some diagnostics, which would be very positive. So that's what my comments are.
Dr. Gundry, I thought you did a really great job. I think out of all the 5 presentations, Glypans was the most visually friendly. And I felt like it had really, each slide to slide, I as a layperson could understand and follow your story. So good job on that. I would say that, I think I lost my train of thought. I had a couple of things. I actually liked that you showed kind of the bigger implications around biomarker testing when you talked about prostate cancer and testing for blood. And as Dennis was just sharing his feedback, I thought, oh, there must be some analogies with even DNA testing and how that's become so pervasive in the home. And that could be a great analogy for you guys to share as you try to draw in more potential partners. In terms of your story, again, you had a very compelling story at the beginning. Obviously, you have your own story. I would perhaps weave that in again at the end or throughout to say, how did this product, how is it helping you with your own disease management and progression? And then finally, my last comment, which is more applicable to everybody, is that so when I worked in a health system, my job was to bring in startups to do pilots and partnerships with. So I was constantly meeting with startups, and my train of thought was, what am I replacing? What is the product that I'm looking at replacing in my current operation? And what effort is that going to take on my end? So when you think about, all of you guys have really obviously amazing, novel, innovative things you're working on, but just make sure you don't get blindsided by what you're replacing, because some folks may not want to replace what's currently on the market, and you'll have a whole set of barriers to navigate around that. So I just want all of you to be prepared for that. And I thought of that with Dr. Gundry's presentation more so.
Yeah, I mean, just a quick comment following up on that, Minnie. You've got a standard of care and you've got a new product. So, what are you doing? Are you iterating? Are you leapfrogging, or are you disrupting? And, you want to size your market and put your product in proper perspective. So when you're iterating something, it could be 100 million. When you're leapfrogging, it could be 1 billion. And when you're disrupting, it could be 1 trillion. So just kind of like, setting context to what's the standard of care and what's your product doing within that realm. And then how far have you gone?
One easy kind of mathematical equation I would give to a lot of startup founders is, if you're pitching to me and you're telling me your product will take 2 minutes of a workflow or 2 minutes of a doctor's time to do. And again, I've looked at mostly operational workflow products, but if you're telling me 2 minutes, I have to go back and say, well, that means I want to be replacing something that takes 20 minutes, right? You have to pitch it to say, currently it takes 20 minutes to do this. Our product is going to reduce that to 2 minutes. And oh, by the way, we're also cheaper, faster, and provide a host of benefits. Then I would listen, right? Then that catches my attention. I'm not going to go back and add anything to a workflow that I already have, because as you guys know, the industry is so bad right now. Everyone's burned out. So just make sure your value proposition, you're including that conversation as well to say, I'm doing it better and this is how.
Great. Thank you so much. I just have on the time, and we have about 5 more minutes before our next panel presentation. So let's move to the 2 final presentations, their comments, and we'll start with Mr. Ray Liu. So on behalf of the Kim team.
I'll be happy to start again. So Ray, you're the classic one. Is it sleep apnea, or is it what you're doing in the operating room? You got to choose one. And so I think, once again, when you're talking 2 markets and 2 different verticals, and it sounds like what you're doing in the operating room with non-invasive sensor for blood pressure measurement and by the way, that's a nice tagline, right? Non-invasive sensor for blood pressure measurement. And you can go into like what you're actually doing and then what the markets are you're going to affect. But what I like to do is lead with one product that's the furthest along and that you've got the most data on. And then at the end of the presentation say, oh, by the way, once we get this moving, we do see some other markets like sleep apnea. So that's it.
Ray, I think you should look at a Swiss company called Acutia, which, do you know them?
Yes, I do. Yeah, we have their device as well. Yeah.
Yeah, they have a CE mark already approved. They're trying, they're selling in Europe and they're trying to come here. So same, essentially similar kind of non-invasive wearable that they have. So just take a look at it and maybe, see how you are better than that particular product. Okay.
Yep. Thank you.
Dr. Liu, and for anyone developing wearables, what I would say is that oftentimes some of the barriers for implementation in a hospital setting is thinking about charging, cleaning, device management. Those are monstrous tasks for health systems on top of already the 1,000 things they have to do. So while sometimes wearables can seem exciting, new, really innovative, they actually create a lot of hardship and extra work in the hospital setting. So just be prepared to answer those questions.
Thank you.
Kwame, I want to give you an opportunity. Any last comments for Ray before we move to the last?
So Ray, I'm really encouraged by the data, and you have a wearable that early data suggests is as accurate or more accurate than standard of care. And also something that you have as proof points from tier 1 accelerators to signal that you're on the right path. So that's my thoughts on your team and the product and your momentum.
Thank you, Kwame.
Thank you so much. All right, comments for Team Vanderslice, Mr. Rob Bent's presentation. Minnie, I'll call it.
Well, I'll jump in again. Once again, I'm looking for the tagline, and it sounded like, activating integrins to boost immunotherapies. And so, just by coming in with, something that can simply set context so that the audience that's relevant can automatically go, oh, what's this guy doing with activating integrins to boost immune therapy? That's kind of how I want to hook my audience if I have a 5-minute pitch. Once again, when the two participants that were telling their personal story today, that's just so compelling when you have a brother or something's impacting yourself. And then you tell how that story's impacted you and you give some specific insight, but it also gives you agency. And it also shows, the passion that you have to deliver this solution. So I really like that. If I was going to say something to Rob, it's like, we should try and turn this into more of a story that we can kind of relate to. You were really going fast. I think it's cool stuff, but I had a hard time tracking the story, but the science was there.
Yeah, I hear you. Thank you.
I would say the same thing. Again, as a layperson, unfortunately, I was lost with slide 1. I could not keep up with the presentation. I had no idea what you were selling, who you were targeting. I just couldn't understand all the scientifics, but that's easily solvable. And like I said before, it's probably worth having 2 versions of the presentation, one scientific and one not. And today, your audience is scientific, so it's what you brought.
Thank you.
Rob,
Yes, we do have a few versions.
Rob, I would say that you have a very interesting technology, and you could have probably cut down on the number of slides and maybe improved the way you're presenting. I think it's more of a scientific presentation than a general kind of thing, which is okay. But when you go out into raising money and all that, you have to have a combination of those two. So, but I think there's a lot of potential, especially during these days, with COVID, influenza, all that. If you really can, expand the immune response, and the small molecule is a chemical composition. Is it new chemical entity or?
It is actually one of the co-inventors is here. It's Dr. Vanderslice.
Oh, okay.
And Dr. Biederer, I should say as well.
Yeah, these compounds were developed at Texas Heart Institute and out-licensed to Seven Hills. And so we're at Texas Heart are working on the cord blood angle, but Seven Hills has done a great job of expanding the indications into the immunotherapy and the vaccine indications as well.
Okay, sounds good. Thank you.
And Kwame, again, I want to give you an opportunity for your comments.
So I know Seven Hills has been quite successful in raising dilutive and non-dilutive capital. So clearly there's there. Generally speaking, there's a discount on the visuals when you're raising $1, $2, $3 million. But when you've had so much money put into a company and you're raising 10 million, the expectation is the visuals are crisp, clean. Even, you spend time on an intern to tell a very seamless story, because the types of investors and their need to really focus is a little bit more acute and higher. But again, Seven Hills, I know the company, you guys have clearly onto something, because you've been able to raise millions of dollars to date. So thank you.
Thanks for the comments. They're well taken. Yes.
Thank you.
Thank you so much, everyone. Pitch deliverers, pitch judges. I thought this was a really productive and valuable session, both for those who pitched and those of us in the audience listening to the pitches. So thank you again.